So Lou mentioned he wanted me to say a little bit about my background, and I will just give you not my entire background, but sort of on the stats side of things. So probably like many of you, I got through a bachelor's degree in an environmental science department and I thought I was sort of done with math because I did my required 1 or 2 courses. And then when I got to graduate school, I was a little shocked that suddenly I needed a lot of statistics. One of the reasons I know as much statistics as I do is because of this guy who was my advisor, Jim Clark. He really is a very quantitative statistical person, and I feel lucky that I was in his lab with all the students that he had, and that I would say that I learned most of my statistics, from classes as well, but I got a lot out of being in that lab and learning from my peers. Went to a workshop, which I encourage all of you guys to do if you can, whatever kind of quantitative workshop besides classes, that can be super helpful. Both of my postdocs, I was shocked to find that I was suddenly the statistical expert, which is a little scary because I kind of knew how much I didn't know. And now I keep trying to learn more statistics. In practice, I often learn more from my grad students and postdocs than I'm teaching them, but that's fun too. All right, so with that, I'll switch over to what I would like to talk to you guys about today. And I'm just going to start with this graph from the IPCC because it motivates a lot of my work now. I'm very interested in trying to understand how climate change influences plant communities. We know climate has warmed in the last 50 or so years, and we know that we expect a lot more. And so what I'm interested in is how is this going to affect plant communities? I'm a plant community ecologist, so what is this going to do to the plant communities that I'm interested in? And here's the study site I work at a lot now. This is Mount Rainier National Park. And, sort of roughly speaking, there's 3 kinds of communities. There's low elevation forests, there's montane forests, and subalpine meadows. And really simplistically, this is what we think is going to happen. This is about what we know, right? As it gets warmer, everything will move up. Of course, as biologists, we know things are a little more complicated than that. It's probably not going to be quite that simple. And what I'm interested in is all those complexities, right? So to what extent is those simple predictions not going to be manifested? So the rest of this talk is going to be a little bit of a smorgasbord of different projects that we've done, and I've tried to focus on the statistical quantitative side of things to give you guys a sense of how we've tackled these 3 different questions that all relate to the complexities of how plant communities might respond to climate change and might not follow that very simplistic prediction that communities basically will or species distributions will just shift up. So the first thing we might want to know is, does climate even determine range limits in the first place, right? If climate plays no role in determining where species are, then you can jack up the temperature all you want, but nothing's going to happen, right? So that's one question we would like to answer. These trees that I work on, and that'll become a little more obvious, they're pretty old, like 400 to 500 years is like kind of an average age. So another question that I have is, sure, and a lot of ecologists have, is sure, climate's going to sort of shift where the optimally, like sort of the geographic distribution of where the sort of the best climate for different species is if climate determines range limits. But we don't necessarily know that they're all going to be able to get there at the rate that climate is warming. So one question we might have is, how fast can populations respond? And then a final question that we've tried to answer, or are still in the process of answering, is, given that climate has already warmed in the last 30 years, have communities already responded? So these are 3 questions that together can tell us something about the complexities of how climate change is influencing forests. And I hope to get to all of them, but given everybody else's track record, I suspect I may not, so we may have to slip through that one pretty fast. Okay, so I'll just tell you a little bit about the study site because it's pretty cool. So we are here, hopefully you know that. This is where Mount Rainier National Park is, up in Washington. And if you're interested in studying climate change impacts on anything terrestrial, this is a really fabulous place to work. I mean, obviously it's beautiful, but more than that, there's like a lot of climate in a small space. I mean, I can basically driving, like 20 minutes, I can cross the equivalent climatic gradient as I would driving from Seattle to Juneau, Alaska. So that makes fieldwork a little easier. So this is a great place to study climate change-related questions. Just a few things. I didn't grow up in the Pacific Northwest, and I'd never worked in the forests out there. So when I first moved out there, I'll be showing you a couple pictures of the things that amazed me and maybe for those of you that aren't from the Pacific Northwest will amaze you as well. So first of all, the trees are very big. So this is my former grad student, Eileen, who I assure you is like an average sized person. She is not this, she's not particularly big or small. Anyhow, so this is sort of an average sized Doug fir. Maybe not, it's on the big side, but it's not that unusual to find trees of this size. So the other thing that is really quite staggering is the amount of snow. So this is up at one of our higher elevation sites. This is probably in March. It's probably like 4 or 5 meters of snow. You can see these trees would usually be standing straight up. You can see that like they're not really. And that's kind of an average amount of snow that we get up at the higher elevation. So this is the part of the climate, right, that's really going to change. There's not going to be as much snow, and this really drives the system. And then at high elevations, you have these beautiful subalpine wildflower meadows that, because snow melts in like July, they have like basically 2 months to do everything. So they bloom really, really quickly because they only have so much time. Okay, so as I said, I mostly work on tree species. I don't know, most of you, some of you may know these, some of you won't, but needless to say, there's 7 focal species that I work on, and they're all, if you're coming from the East Coast, they all sort of look the same to you at first because they're all conifers, but they are different, and you can identify them with time. Okay, so let's start with how does climate even play a role in determining range limits? So the approach we took to this question is we have a number of species. They occur at different elevations, so here's sort of an average high elevation tree species. As you go up the mountain, there's an upper range limit and a lower range limit. And if climate determines those range limits, we might expect to see a pattern like this. In other words, at this upper elevational range limit, we might expect that if climate is sort of preventing these populations from expanding, that we would see for these individuals, if we were able to jack up the temperature, that performance or some measure of fitness would increase, right? So that could be year-to-year variation, or it could just be local warming, but we would expect that these individuals would benefit from warming if climate determines that range limit. We would expect a similar relationship between performance and climate at the lower range limit, just in an opposite direction, or maybe with a different climatic variable, right? So similarly, if climate determines the lower range limit, we would expect that these individuals, if they're really stressed, for example, by warm temperatures, that if you jacked up the temperature, their performance would go down. So that's one way to get a sense of whether climate plays a role in determining range limits. So that's what we did. We went to 9 different stands, like we basically hiked up the mountain from low elevation to high elevation. We went to 9 locations. These curves are the 6 species for which we assessed performance, and I'll tell you a little bit in a second about what our metric of performance was. Just to persuade you that climate did change a lot across that gradient, here is how mean annual temperature declines and winter precipitation increases. So we have these 9 locations, and you can see they cover at least the upper, you get close to the upper elevational range limits for all of them, and the lower for a couple of them. So what did we measure? Well, trees are basically very inconvenient for almost everything demographic because they live so long. But one really nice thing about trees, one thing that makes them convenient, is they lay down a record, or at least temperate trees do, they lay down a record of their past growth. And we can take advantage of that, so we don't have to wait around for 50 years to see how they respond to annual fluctuations in climate. We can core them and go back in time. Fortunately, at Mount Rainier, there are long-term climate records going back to the 1920s, so we can line up annual growth with climate and ask to see, do we see those patterns for these tree species at upper range limits? Do we see greater climate sensitivity at the range limits? And if so, is it in the direction that we would expect if climate was preventing these populations from expanding? Okay, so that's what we did, and I'll talk a little more about exactly how I did that in a second, but here's an example of, for one species at its upper range limit, what's the relationship between mean annual temperature and growth. And so each of these dots is a different year of growth. And so this is the temperature experienced by that group of individuals, and this is a measure. I'll talk a little bit about what standardized means. And the main thing I want you to take away from this is, yeah, they're scattered, but in general, as it gets warmer, these trees grow more, okay? And that's exactly the signal we would expect at the upper range limit if climate was determining range limits. Okay, so what's going on at the lower range limit? We'd expect maybe an opposite relationship or maybe a different climatic variable. But in fact, what we see is absolutely no relationship between climate and performance. And I mean, I'm just plotting up mean annual temperature here because it's convenient. We tried, we did the model selection side of things. We tried a lot of different climatic variables. Nothing we could look at would make the species sensitive to climate at its lower range limit. So this is just one of the species. What about, I told you I did this, and that we did this analysis for 6 species. What happens when we look at the other 5? So I'm just going to summarize in a table here. So this is what we would expect to find. We would expect at the upper range limit to find a positive effect of warm temperatures or growing degree days, and a negative effect of snow, because the higher you go, the more snow it is. If climate were determining range limits, then we'd expect the opposite coefficient. So we'd expect basically that temperature has a negative effect and maybe snow has a positive effect at lower range limits. So I'm just going to use this table to summarize what we find for species, and basically this is the hypothesized relationship, and all you have to do is look and see if it matches. So here's what we find, and these species are arranged by their basically their elevational limit. So these are this is the highest elevation species, and this is the lowest elevation species. And the main thing I want you to take away from this is these arrows all line up with our hypotheses for these 3 high elevation species, but basically no other range limits, basically performance at all these other range limits, both the upper range limits for these low elevation species and the lower range limits for these high elevation species, none of them match up to the hypothesized prediction. So that just means that some species are sensitive to climate at their range limits, suggesting that climate determines their range limits, but not all species, and we're still trying to work out what's going on for those other species. But now I want to spend a little bit of time on how we actually got here and some of the challenges we faced from a statistical point of view. So the first thing is, and I'm sure a lot of you that have worked with environmental data or climatic data have probably faced this. So, we had this long-term climate record, and we could distill. I mean, we had monthly temperature and precipitation data, and we could turn that into a number of different things: growing degree days. We could use calculations to turn that into potential evapotranspiration, like a measure of drought. We could turn that into snow duration, amount of snow. We could turn that into all kinds of things. But then when you look at the sort of pairwise correlation between all these explanatory variables, you see that there's a lot of climatic variables out there that seem like they could affect tree growth, but a lot of them are correlated, right? So for example, mean annual temperature and growing season temperature, potential and growing degree days and mean annual temperature. Those are both plausible variables that might affect tree growth from a biological point of view, but you wouldn't want to stick them in a model together, right, because they're highly correlated. So the first thing we had to do is come up with a way to basically not put in, we explored our explanatory variables and came up with a model selection framework to essentially not put in climatic variables that were highly correlated. And that also obviously affects our inference. If we find a strong relationship between growing degree days and growth, what we know is it's sort of energy/temperature related variables. It's not necessarily growing degree days. It could be any number of other things that it's correlated with. So that was one thing we had to do. The other thing was, we had climate at one location. So this is a map of basically the distribution of mean annual temperature in Mount Rainier National Park from PRISM, kind of smoothed. So we knew what climate was here, but our stands were at all these other different locations, and we wanted to use at least our best estimate of what the trees were experiencing where they were growing. So essentially we had to go to some sort of downscaling. So we used lapse rates that we also validated with these microclimate sensors that we had to basically take this temperature record that's measured at one location, and we did the same thing for precipitation. That was a little harder to validate, to turn those into explanatory variables that were at least as close as we could come to relevant to what our trees were actually experiencing. And then the final thing, this gets a little bit into sampling as opposed to actually necessarily statistics, but for those of you that have worked with dendrochronological data, you know that a lot of the statistics that you read about in sort of the classic books, they were developed by folks that were interested in predicting climate from trees. I don't really care. I mean, I didn't really want to do that. I wanted the trees to tell me how they felt about climate. And so what that meant is we had to think hard about those standard techniques and sort of think about, are they really going to do what we want them to do? So standardly, if you're building a chronology and trying to predict climate from trees, what you would do is you would go out and you would only pick climate-sensitive trees. You'd say, this tree has no other trees around it, and it looks like it's in a really crappy soil, and that means it's probably going to be picking up climate. Well, you know, that means that, you might be getting a good climate signal, but you're not necessarily getting the average tree. The other thing you would do is you would core 20 of these, and then you would average out all that annoying stuff, right? Like the branch that fell on the tree 20 years ago or the insect outbreak that might cause it to deviate. So we didn't want to do that because we were actually interested in that other annoying stuff. So basically what we did is we went out and cored random trees. I mean, we did have ways of picking them, but what I mean is we didn't select trees that we thought would be more sensitive to climate. And we used a mixed effects models. We used random effects because we were interested in the individual, like the individual to individual variation and how they might be sensitive to climate. We didn't sort of care about the average tree. We did care about the average tree, but we also wanted to know what the variation was among individuals. Okay, so where are we going with this in the future? Like another thing that really bothers me about this analysis, which we didn't do anything about, was sort of standard dendroclimatology. You sort of take a spline and you take out the size. Like as trees go from little to big, the ring widths vary just because of their size, right? Because ring width doesn't translate, like a width measurement doesn't translate to basically the volume of wood that the tree's putting on. So one solution that a lot of dendrochronologists do is they take a spline to that data and sort of average that out. That kind of annoys me as well, or the problem with that is if you have a long-term trend, right, like climate change, then you might be averaging that out as well. And I also think we can do better because we actually know how trees grow. We have some idea of the relationship between size and growth. So one thing that would be nice to do is to remove the size growth trends with some sort of rather than just a flexible spline, come up with some model of how we think ring width varies with size, or at least an empirical relationship. There's lots of permanent plot data out there that we could at least take out some of these relationships in a less sort of fuzzy way. And then it would be nice, rather than doing this, like running the analysis for each stand individually, it'd be nice to build a model that does it for all the trees at once, so we can get an estimate of climate sensitivity across the entire climate space of what trees are experiencing. Okay. So then the next question, how fast? So climate does determine some range limits, especially at upper elevations. So now we might be interested in, when climate changes, especially for those tree species that do seem to have their upper range limits, like sort of potentially determined by climate, how fast, are they going to be able to respond fast enough? And so how might you answer that question? Well, the thing to recognize is that trying to understand how fast range shifts will occur is fundamentally a demographic problem. So if you have a climatic gradient NIMBioS species, let's do that again just because I spend a lot of time on that custom animation. So climatically suitable space, climate gradient, climate shifts, and the way for the actual range of the species to shift is populations have to grow at the leading edge and they have to crash at the trailing edge, right? And so range shifts could be slower than the rate of climate change if there's slow population growth here or slow sort of population extinction at the leading edge, at the trailing edge, right? So that's an explicitly demographic question, like how fast can these populations grow? Okay, so how did we do this? So you could use, we actually have a lot of data at the seedling stages, at the sapling, that's the sapling stages, and at adult trees. So you could use matrix models. Caswell has an entire book on it. But because we actually chose to use integral projection models, which are a relatively new analytical tool, and there's lots of great resources out there. There's IPM-PAC and R package. There's, I think that's actually Metcalf 2014, but there's a number of papers that have come out where you can essentially relate vital rate, so growth, survival, and reproduction, to the size of your organism. So this is great for organisms like trees, where they're sort of they get bigger and bigger, and probably their growth and survival and reproduction changes in a continuous way, and it would be hard to sort of come up with it would be very subjective to come up with a matrix with these little categories, right? We think these happen in discrete ways. So we chose to build an IPM. for one of our trees. And the great thing about integral projection models, much like matrix models, is you can pull out a lot of metrics that are presumably of interest, so population growth rates, but also what's the stable age or stage distribution, which of these like size classes is most determining lambdas, what's the sensitivities and the elasticities. And you can also, once you have this model, it's like a hypothesis, you can play around with it, you can use simulations to explore different scenarios, and you might potentially be able to look at transient dynamics. Okay, so what's the data? So we essentially focused on our most abundant tree, and we have data from these 15 stands, but AB's amabilis only occurs at 11, so we have growth, mortality, and reproduction at 11 locations that sort of cover the altitudinal range, because we wanted to know is population growth rates potentially higher at the upper range limit, like a signature of recent climate change. How is that varying across the range of the species? So that's where we collected our data. The data we collected, so we have seed production. These are not laundry baskets. They're seed traps, and they collect seeds that are then collected and counted in arduous detail. These are two undergrads that are counting little seedlings and tagging them. So we have information on what happens at the early life history stages. We have some, although less than we would like, information of what happens to saplings, so it's sort of the intermediate sizes. So this is Sarah, who's harvesting a sapling. This is permitted, by the way, for those of you who work in a National Park Service. That permit took a lot to get. And when we actually age these trees, so these are probably about as tall as I am, like you start to get a hint that these trees might respond very slowly, because this little guy is probably about 80 to 120 years old. We stopped calling them seedlings when we realized that, because I was like, I can't call something that's like 3 times as old as me a seedling. Okay, so we have sort of the intermediate stages, and then we're really fortunate to have 35 years of growth and mortality in these permanent stands, because they're ones that Jerry Franklin, an ecologist, set up. So basically remeasurements every 5 years of size and mortality. And again, start to get a hint that things might happen really slowly, because it's about 1 to 3% annual mortality rates maximum, and usually a lot less than that for these trees. Okay, I'll jump right to the results. And I apologize, this graph is relatively recent and probably will change with the next formulation of the model we put together. So it's not as nice as I'd like, but what you see here is across the elevational range of the species, the closed dots are the population growth rates of the species at the stable stage distribution, and the open dots is if we take the current stage distribution and basically apply the matrix that we built to it for a couple of years to try to, because we weren't sure whether the stands were actually most of them are not at stable stage distribution, even though they're old growth, which is interesting in and of itself. The main things to pull away from is the lambdas are all, like, they're pretty close to 1, but they're all below 1, but they're not very high, right? Like this is like a 0.99 or a 0.97. So these are a lot higher, but they're still, these lambdas, if you were working on an annual species or on like herbaceous perennials, these aren't very high lambdas. So these populations are probably not going to be able to respond very fast. I mean, they will be able to respond very fast if a disturbance comes through and kills everything. So on the mortality side of things, things could happen really fast. But on the growth and expansion side of things, it seems pretty unlikely that they're going to be able to respond particularly fast. This is still preliminary. We're still working out the kinks in this model. So a couple of things. We had a lot of problems. I could have spent the rest of the talk talking about the problems we had, because it's pretty complex, but I'll just talk about a few. So one is we had a lot of implausible data, which probably most of you guys have faced. So when you're looking at growth data, it's true that seedlings can decrease height from one year to the next due to herbivory, but we saw a lot of decrease that we were pretty sure was not due to herbivory. So we had actually a fair amount of negative growth rates, also in DBH, which does size, which doesn't make a whole lot of sense. So we had to come up with models that had measurement error in it. So that was one challenge. Of course, we have like random effects coming out of our ears. We have individuals that are remeasured. We have stands that are remeasured. We just have a ton of random effects, so we put a whole bunch in there. And then the final challenge we faced was actually in putting this, because essentially what we're doing is we're parameterizing an IPM, and we're fitting a bunch of vital rate models, hierarchical Bayesian vital rate models, to parameterize it. And actually, that was harder than we thought because we had basically data collected at these 4, even though we wanted to model this continuously, we had sort of tree level data that was for 35 years, every 5 years, where the metric of size was diameter. We had saplings where the metric of size was height, and we had seedlings. Like, these are 3 different collection efforts just because of logistics. And so we started out by creating a piecewise IPM. We had like a different model for each one. That wasn't very satisfying because, like, for example, our survival for our tallest, for our biggest seedlings right before they transitioned into being a sapling would, for example, be low. Like, there were these funny jagged things in our, so the you expect as the tree gets bigger, just from biology, that survival rates would get better. But we would have lower survival predicted for our largest seedlings than for our smallest saplings, if that makes sense. So these were all problems, and what we went to, and we have I don't think anybody else has done it this way, maybe because it's crazy, is we basically translated everything, not a seed, actually, fortunately they don't seed bank, so they turn into first-year seedlings. We turned everything into diameter at ground level. So the tiniest seedling has like a 0.5 millimeter diameter at ground level. And that's so that we could model size the same for everyone. And that meant translating diameter at breast height to diameter at ground level and height to diameter at ground level, which is a whole other mess that I won't go into. But that's the choice we made because then we could model size continuously. So we would like to apply this to other species. And I should also say the other thing we would like to do is make sure to get this model right, because I'm still, I don't know about this. I'm still not 100% sure that we're doing this right. I think it makes sense, but I guess what I would say, what I'm not sure about is if some of our crazy assumptions that I've seen nobody else make, influence our results, right? Influence our conclusions. So we have some playing around to do to figure that out. All right, so climate does determine some range limits. Doesn't seem like these populations can respond particularly fast to climate change. The final question, and I think I have time for this, is basically, given this, we might expect that things won't change a whole lot or not very fast with climate change. But of course, one great way to answer the question of how rapidly communities can respond to climate change, if you have that luxury, is to go to the past, right? To ask, have communities already responded to climate change? So let's think about what it means when, let's think about a perspective that we might take about how communities would shift. So I talked a little bit about how we expect all species and therefore communities to shift up. And the reason that works is, here's an elevational gradient and a climatic gradient. Here's like a nicely ordered set of species that all have their own climatic optimum. And so at any one location, we have a community that's made up essentially of the species, that are able to occur there and the relative abundance according to how far they are from their climatic optimum. And with climate change, we expect all these species to very well behaved march up. And so if you were standing there, you would see the community around you change in a very predictable way. Of course, we might expect nothing to happen, right? Because these populations are really slow to respond. Or we might expect species to move in different directions and communities to sort of reassemble. So we were interested in that question. And one of the reasons we were interested in that question is because we actually have 35 years of data for 15 communities, for 15 tree communities. So these communities were set up in these plots were set up in 1978, and they've been monitored every 5 to 6 years since then. And so we have a community in 1978 and in 2013, which was the most recent census, and we can ask, there's actually been about 1/3 of a degree of warming in the Pacific Northwest since then. So we can ask, have these communities already shifted? And we wanted to compare the changes we saw in composition to the ones, to be able to test different or think about different hypotheses, like is there no change? If there is change, is it what we would expect from warming? So these are our communities, and essentially through differential mortality in growth and growth, have these communities changed? I should say that hopefully it's still obvious, although I feel more, this job makes me feel more haggard every year, but hopefully it's still obvious that in 1978 I wasn't out there measuring trees. This is, Jerry Franklin set these up, and these are some of the folks that have been instrumental in helping remeasure and archive the data. So we have these tree communities we follow over time, but we need an expectation, or I wanted an expectation. Maybe they've shifted, but how do we know, have they shifted according to expectation? Where does that expectation come from? So one thing you can ask, if communities move upward, the implication there is that spatial turnover at the time when those communities, were that you started monitoring those communities would predict temporal turnover, right? So as you go, if you're at this location on the mountain and you want to know what's going to happen in 35 years with 0.3 degrees warming, you would essentially walk downhill the equivalent of 0.3 degrees and look at the community there, and that would be your expectation. So the great thing, and it's a little more complicated than that because there's actually more than just temperature that varies, but essentially that's the prediction we would have. And the great thing is we actually have data from around the same time. We have close to 500 plots that were established at a bunch of different locations that across these climatic gradients that allow us to essentially look at how community compositions varies with climate and generate that expectation. So it was really fortunate that those were collected right around the same time. So we need a way to turn that community composition into, as a function of climate, to turn that into a spatial prediction, to turn that spatial gradient, the spatial turnover into a prediction that we can then ask, have our 15 communities followed that? So what we did is a canonical correspondence analysis. This was like the first multivariate analysis I ever did, and it was, I felt like it was too easy. It didn't take me very long to generate this, and I don't know how much I believe it. But so what this is, canonical correspondence analysis, is a multivariate technique where you can take environments we had for each of those plots, for each of those 500 plots. We knew we could determine what the climate was from these PRISM climate models. And we have community composition, in this case basal area. And we can line those up, and through the magic of multivariate techniques, it turns it into a plot like this. Each of these dots is actually one of our plot samples. These are some of the species, and this is basically telling you where in this sort of dimensionless space these species occur. And each of these arrows represents the climate gradients, the environmental variables that we put in there. And the arrows just tell you that in the left-hand side of this graph, it gets warmer. In the sort of down here, it gets snowier, and down here, it gets wetter. That's just what that's telling you. And I mean, again, I don't know. I'm not 100% sure if I'm satisfied with this method, but what is the case for those of you that know these species is it does actually, those predictions are reasonable based on what we know about the larger biogeography of these species. So for example, here's two congeners, Sitka spruce and Engelmann spruce. Sitka spruce occurs across the by the Olympic Peninsula. It occurs warm and wet, and that's the place it comes out. And Engelmann spruce, cold and dry, and it comes out in the cold, dry region. Here's mountain hemlock and western hemlock, which are congeners, but this is a high elevation and a low elevation species, and this occurs in the warmer, and this occurs in the colder side of the space. Okay, so how did we use this? So here are our 15 stands in that same canonical correspondence analysis space as they were in 1978. Each of these arrows represents where we would predict them to move in canonical in this space with 0.32 degrees of warming, right? So what I'm going to do now is I'm going to plot on where they actually were 35 years later, and if they've all shifted according to what we'd expect to see all those black dots line up right at the end of where these arrows are. And that is not what we see. Another way to look at this is to sort of turn these angles, like put them all at the 0 degree. Here's the expectation. Here's where all those arrows go. I mean, some of them actually move surprisingly far, right? Like, in other words, there's been shifts in composition. That's usually when an enormous tree dies, I have to say, because that changes the basal area a lot. But most of them, it's not like all of them are moving in the same direction. So we have change, but it's sort of idiosyncratic. Okay, so that's where we've gotten with that. I actually do want to do a more sophisticated or a different kind of analysis, and that's a joint species distribution model. For those of you that are familiar with species distribution models, that takes it one step further by actually modeling the relationship between species distributions and climate jointly, and at the same time modeling the covariance between species. There's two nice papers, and the Pollock paper actually includes a bunch of OpenBugs code, so that can be really useful for those of you that want to implement that. You get some really nice information out of it, like which climatic variables most strongly predict community structure, and where is the sensitivity to climate most amongst your communities. So these aren't my graphs, so these are from the Clark et al. Paper, but that's something we want to do in the future. I think I have 5 minutes left, which is great. So I'm going to end with sort of my ecological conclusions, and then I'm going to also give you sort of my 2 cents / suggestions, because we're supposed to be doing that. So, okay, we started with a simple prediction that plant communities simply move upwards, and I hope that I've convinced you by answering these questions that that simple prediction seems unlikely to be materialized. Probably we're going to have lagged and idiosyncratic range shifts. Exactly what those are going to be is a little harder to predict, which I think actually makes it a lot more of an interesting from a purely intellectual question. I hope I've also showed you a little bit, given you a little bit of a sense of the types of statistics we've used to answer these questions. So now I want to spend just a little time giving you, I guess, my 2 cents. I mean, it feels very pedantic and professorial, but I guess that's why we're here. So I've used a lot of different approaches, like all the way. I've used some simple regressions. I've used some hierarchical Bayesian approaches and some more complicated approaches, and ones that are sort of in the middle. And to me, there's often this trade-off between how easy, and I don't mean easy in a derogatory sense, but like how quick it is, and how easy it is to implement this, or how fast it is to implement it, and know that what you're getting is reasonable given the statistical assumptions, as opposed to having to code all your stuff and not even being sure if the code is right. Versus flexibility and complexity, right? A t-test regression and ANOVA, you can go to R and just type in AOV, LM, I think it's t.test, I don't know. It's not that hard, and those packages have been well tested by, you don't have to worry that the code underlying it doesn't work. However, they're not very flexible, and they don't accommodate a lot of complexity, and a lot of the biological problems we deal with are complex. So I'm not somebody who feels like everybody needs to do this. My suggestion would be use the tool that works. Like, you can use a saw for some tools. You don't need a table saw for everything. Don't be afraid to use a simple tool if it's the right tool to answer your question. And don't forget to ask yourself, what is the question? Because sometimes that can help you decide what the tool is. I will say, the flip side of that is, if you don't know what tools are out there, you may be using a really simple but inappropriate tool when you could be using a more complex. And I don't know if more appropriate is right, but that makes it sound like it's sort of a grind, like, we just have to do this because it's the right thing to do. But you can also get a lot out of the more complex tools. You can get a lot more information and answer a lot more fascinating questions. So don't forget that. Think about the stats before you collect the data. It's always good. And really have fun. I mean, start simple, work up to the complex. Use that complex tool if you have to. If you're just doing it because you want to, then that's fine if you want to learn the tool, but at least be aware that that's what you're doing, and you actually maybe already have the answer and could start writing your paper on the side of writing papers. And you can get advice from other people on that side of things. I have a bunch of other data that I didn't have time to talk about, but for any of you that are interested in phenology data and citizen science data, I have a lot of that kind of data. I've done a bunch of dispersal modeling from this kind of data. I have a whole other set of research I do on annual plants, which is basically using demographic data to fit coexistence models. I'd be happy to talk to any of you about any of those if you're interested. This is my first field crew in 2008, and this is my 2013 field crew, so that's very exciting. 7 years of data. Definitely all the data I presented would not have been collected without these guys. And these are just some snapshots of my lab and various funding sources. And with that, I think I have 1 minute for questions, or maybe 2.